Hi, in this episode I'm going to discuss the specific tissues that glycolysis takes place in. Not all of them, but most of them, and I'll give you a good idea of what it's for. So, I'm going to start with the liver. The liver undergoes what we call hepatic glycolysis. Hepatic just refers to the cells within the liver, anything to do with the liver. And it uppers blood glucose levels, keeps them nice and low down compared to prevent you going hyperglycemia. Hepatocytes derive their energy from molecules other than glucose, for example fatty acids, and if glucose is converted to triacylglycerides in the liver, which is stored in adipose tissue, so basically these are fats, it can also lead to fatty liver. Next time we're going to discuss the eye, and the corneal lens in particular, and as you'd imagine, the cornea and lens are both devoid of blood vessels. They don't have any blood vessels, because they need to be transparent so you can see through them. If you had blood vessels, you'd have that across the front of your vision. They have no mitochondria either, so they have to undergo anaerobic glycolysis. They can't complete the pyruvate dehydrogenase complex to acetyl-CoA. And they obtain the glucose from the actual vitreous body and aqueous humour of the eye, that's the bit inside of it. As such, that's how they operate. Going to talk about the kidney medulla, as you can see. The medulla here are the darker bits, and these actually have quite a poor blood supply. Now, I know it looks like they have a good blood supply from the renal artery, but that actually goes into the cortex and then back out by the renal vein. However, these medulla have a rather poor blood supply and as such need to undergo glycolysis, and they also have a high ATP demand, so they need to do glycolysis at a high rate. An interesting one is neonatal tissues. Neonates, generally they're dependent on glucose and ketone bodies in pregnancy. However, during birth and placental separation they can be hypoxic for like 30 minutes, which is a very long time. Normally you want to keep hypoxia under 1 to 2 minutes. So for 30 minutes to survive that is an incredible feat. During these hypoxic conditions, anaerobic glycolysis supplies all of their ATP needs, so they manage to run purely on anaerobic glycolysis for that time. And during the last stage of pregnancy, hepatic glycogen concentration is actually at its highest in the liver, so it allows for the release of lots of energy from the liver into the bloodstream during birth to maintain this anaerobic glycolysis that they need. Erythrocytes, blood cells, they have no mitochondria and rely on anaerobic glycolysis for energy, as a lot of things. The glucose 6-phosphate produced by hexokinase is required for glutathione production. This is one of the alternative pathways mentioned before. And glutathione is a reducing agent that prevents oxidative stress by attacking oxidizing agents, preventing them affecting other parts of the body. Production of 2-bisphosphoglycerate is also linked, but you don't really need to know that. It just means skip a step producing ATP on the pathway to the 3-phosphoglycerate. Tumour cells also undergo anaerobic glycolysis quite often if they've grown around the blood vessel or can't get blood supply to the peripherals of the tumour, or just haven't grown near blood supply. Generally they tend to be hypoxic, poor blood supply as I just said, and they produce HIF1, which is hypoxia inducible factor. This increases the expression of GLUT1 and 2. Do you remember that earlier we mentioned those were membrane proteins designed to transport glucose into the cell as to allow for glycolysis to happen. And it also increases glycolytic enzymes, which are the enzymes used in glycolysis. It also indirectly causes angiogenesis, which is the growth of blood vessels into the tumour. Now, what the current chemotherapy treatment, not current, but theoretical chemotherapy treatments are thinking about targeting HIF1 as a possible way of making so blood vessels stop growing into tumours and slow down the growth, and overall allow chemotherapy to be more effective as it slows down its growth, preventing it bouncing back from the chemotherapy treatment. Anyway, that's it for this episode. Next episode I'm going to discuss the diseases relating to glycolysis and the PDH complex. Yep.